Assalamu alaikum wa rahmatullahi wa barakatuh. Today we are going to continue our gastrointestinal tract lecture, and also we are going to start gut associated gland. Today we are going to continue with rectoanal junction and gut associated gland. We are going to discuss one of them, which is pancreas. Now let's see our internal learning outcome of our lecture today. By the end of this lecture, you will be able to list the histological structure changes that occurring at the rectoanal junction. You will be able to identify one of the gut-associated gland, which is the pancreas. From the pancreas, you will be able to discuss the structure and correlated function of the exocrine portion only of the pancreas. Now, let's start our lecture with talking about the rectoanal junction. Rectoanal canal or rectoanal junction, we are going to discuss the region of transformation from the rectum into the anal canal. I have to memorize to you that rectoanal canal is considered as a tube. So in order to talk about part of the tube of the gastrointestinal tract, you are going to talk about 4 layers or describe the changes that occur at the level of the 4 layers. What are they? The first one from inside to outside is mucosa. The second one is submucosa, which is a dense connective tissue coverage. What after them? They are the muscle which is responsible for peristalsis, which is called muscularis externa. And finally, we are going to have serosa or adventitia. Here, we are going to talk about rectoanal canal or rectoanal junction. So it is not covered by peritoneum. So we are going to have a layer called adventitia. What about mucosa of the colon? Mucosa of the colon was, as part of any tube, mucosa will be formed of 3 parts. What are they? They are epithelium, lamina propria, and finally muscularis mucosa. Epithelium of the colon was showing a structure called crypt, deep invagination into lamina propria, and it is called crypt. Okay, this was I memorized you what we say in the previous lecture. Now let's go to talk about the rectoanal. Rectoanal canal is a direct continuation of the rectum to go to the outside through anal canal. The mucosa of the rectoanal canal anatomically will show us a longitudinal fold. This longitudinal fold will called column of Morgagni. Do you remember circular fold of the mucosa? In the small intestine, it was called brachicircularis. Here in the rectoanal canal, we are going to just have a fold of mucosa of the rectoanal canal. This will make a longitudinal fold. This longitudinal fold would be called column of Morgagni. Adjacent column of Morgagni due to longitudinal fold will be interconnected by a membranous fold forming what is called anal valve. Let's see a picture for this. Here we are going to have a valve. This valve is called a membranous. This is a membranous fold communicating a longitudinal fold which is called column of Morgagni. This membranous fold is forming a valve. This is called rectoanal valve. Let's talk a longitudinal section to see what happened to our 4 layer as we are changing from the rectum into the anal canal. Microscopically, to identify the changes that occur at the rectoanal junction, it will occur as follows. The first layer to talk about will be to see the changes which occur in rectoanal canal. In rectoanal canal, we are going to have a site which contain the rectum. Here we are going to have a site, our femoral side, which is containing the rectum, which is part of the colon, or sorry, part of the large intestine. The second part will be go to the outside, which will be the anus, and here we are going to have the rectoanal canal. As regarding to talk about the difference that occur from the rectum to the anal canal, we are going to take each layer separately and see what happened. The first layer from inside to outside will be mucosa. Mucosa will be formed of 3 parts. Do you know the 3 parts of the mucosa? Yes, the first part is the epithelium. Here, this is our epithelium. The second part is the connective tissue covered, which is the lamina propria. And finally, we have the third layer of the mucosa, which is muscularis mucosa, which is smooth muscle fiber arranged as inner circular and outer longitudinal. Let's see what happened to each layers of lamina propria. Let's see here. In the rectum, we were seeing that the epithelium was showing deep invagination. This deep invagination was called crypt. This terminal epithelium was called crypt. What is the lining epithelium here? The lining epithelium here in the rectum was simple columnar epithelium, as we mentioned before. Can you see here what happened to the crypt? Crypt come here to add the anal valve to be changed into another type of epithelium with disappearance of crypt. Again, here in the rectum side, we was having simple columnar epithelium which was forming a crypt, and this crypt of the rectum was shorter and fewer and became disappear where? At the anal valve, in this region at the anal valve, where it is changed into another type of epithelium. Can you see the second type of epithelium which appear here? Is it simple columnar like that? No, it is more than one layers. Here it is more than one layers. So it is simple or stratified? It is stratified. It is stratified squamous. Is it keratinized? We are inside the body. It is non-keratinized. So here we are going to have at the anal valve stratified squamous epithelium non-keratinized. In order to be changed at the anus into another type of epithelium which is stratified squamous epithelium keratinized. Stratified squamous epithelium keratinized is considered epithelium of the skin. So we are here at the anus and the epithelium started to be changed into skin by its appendices. What is meant by that? It means that this is skin, so it contains hair follicle with its sebaceous gland. It contains a modified sweat gland, modified sweat gland, and this is the skin side. So this was as regarding epithelium of the mucosa. What about lamina propria? Lamina propria would be continuous. Here it is lamina propria of mucosa. Here it is lamina propria of the epithelium. Here it will be changed into another name from lamina propria to be changed as we have skin to be changed into dermis. Okay, the third layer of the mucosa is considered the muscularis mucosa, and it will be continued with us till it will be at reaching the anal valve. It will be abruptly or disappear. It will be subdivided into one bundle and then disappear. So which layers of the mucosa disappear? It is muscularis mucosa. Which layer changes from simple columnar into other type? It is epithelium, which changed from simple columnar epithelium to be stratified squamous epithelium non-keratinized at the anal valve, and finally at the anus it will be stratified squamous epithelium keratinized, which is skin with its appendices like hair and sebaceous gland as the modified sweat gland. Let's see this changes written. We talk about muscularis mucosa as far as we reach the rectal column or anal valve. It were subdivided into bundle and then disappear. Now let's go to talk about the submucosa. Submucosa, after disappearance of the muscularis mucosa with the lamina propria, will be the same part, and it will show some, and it will contain some dilated blood vessel. Some dilated blood vessel was considered as a region of hemorrhoid, and this hemorrhoid is called internal hemorrhoid. So here, after disappearance of this layer, the connective tissue of lamina propria became continuous with that of submucosa. Both became the same layers. But here, at the level of the anal valve, we are going to have dilated blood vessel. This dilated blood vessel is called internal hemorrhoid. Why internal? Because we are going to see another dilated blood vessel, but to the side of the anus, and here it is called external hemorrhoid. So some mucosa at the level of the anal valve contain dilated blood vessels, which are called internal hemorrhoid. While at the anus side, it became another group of dilated blood vessels near the anus outside, and this is called external hemorrhoid. Let's see and see what happened at the third layer, which is muscularis externa. Muscularis externa is arranged as inner circular and outer longitudinal. Let's see what happened to each layer separately. At each layers, the inner circular will be thickened to form the internal anal sphincter, which is involuntary sphincter, while the outer longitudinal smooth muscle fiber will extend between the internal anal sphincter and skeletal muscle fiber to the external sphincter to be lost. It will be faint gradually. So the outer longitudinal muscle fibers will not form anything. It will passing through the circular smooth muscle and the skeletal muscle, which will appear later in order to form the voluntary external anal sphincter in order to be lost in the dermis. Let's see this what happened on a photo. Here is a photo of the all layers which occur. Let's see or memorize what happened before. Here we can see that we was having epithelium of the mucosa, which changes at the anal valve into another type of epithelium, which is stratified squamous epithelium non-keratinized, and at the anus it will be keratinized to form skin with the hair and sebaceous gland with modified sweat gland. We are going to have lamina propria, which after disappearance of muscularis mucosa, it will fuse with submucosa, which at the anal valve will contain dilated blood vessel called internal hemorrhoid, and at the level of the anus it will contain another dilated blood vessel which is called external hemorrhoid. Let's go to the muscularis externa, which is formed of inner circular and outer longitudinal smooth muscle fiber. This is the inner circular and this is the outer longitudinal. What happened to the inner circular here at the level of the anal valve? Can you see? It became thickened. Why it became thickened? To form the involuntary anal sphincter. What else? The external longitudinal or outer longitudinal smooth muscle fiber. Can you see? It passes in between the smooth muscle fiber above and the skeletal muscle, which starts to appear to be divided into bundle and disappear gradually. Here at the level of the anal valve start to appearance of skeletal muscle fiber. For what? Why it is skeletal? Skeletal muscle, as you know, it is a voluntary muscle. So this will make the external anal sphincter, which is voluntary sphincter. Okay, what about the fourth layer, tunica adventitia, and no changes appear in tunica adventitia. It is just connective tissue continue. Okay, let's see a clinical application about the dilated blood vessel. Dilated blood vessel was found in submucosa, as we mentioned here. We are going to see here this dilated blood vessel, one internal and one external dilated blood vessel. Okay, this is both blood vessel, dilated blood vessel, was seen in submucosa after disappearance of muscularis mucosa. We have one near the anal valve, it is called internal hemorrhoid, and another one nearing the anus, which is called external hemorrhoid. This blood vessel may increase in size, forming a clinical condition which is called bile. It is manifested by a very painful defecation, very painful condition on defecation with appearance of fresh blood during making stool. It may be of two types. If it is congestion from the inner one, it is internal hemorrhoid. If it is from the external one, it is external hemorrhoid. Okay, so what is the clinical condition arising from congestion and dilatation of blood vessel of the submucosa? It is a condition called bile. It is increase in the vessel of the submucosa of the venous plexus, which will lead to a clinical case called bile or hemorrhoid. Both are the same name. It is manifested what? Will the patient came complaining from, it will complain from very severe pain during defecation and appearance of fresh blood with defecation due to engorged blood vessel, which may be ruptured. It may be of two types. If it came from the internal hemorrhoid, it will be internal. If it came from the external one, it will be external. Okay, and now let's talk about the second part of our lecture today, which will be about gut-associated gland. Gut-associated gland is the gland associated with our gastrointestinal tube in order to help in performing the process of digestion. What are the gut-associated gland we have? We are going to have two major glands. One of them is the liver, and we will discuss in the coming lecture, and today we are going to discuss pancreas. Pancreas is considered as a large gland. It is present in the C-shaped structure of the duodenum. We see its head. Pancreas is considered as mixed gland. What about is a mixed gland? Mixed gland means that it is both exocrine and endocrine gland. What is mean by exocrine? It means that it releases its secretion into the its secretion through tube. And endocrine gland, it means that it releases hormone, blood hormone, to be released into the circulation through blood vessel. Both of them, exocrine and endocrine function, so are performed by different cells or by different structure. Now, pancreas, as regarding to us, is considered the largest gland of the body after the liver. What are the histological structure of the pancreas? As you know, it is a parenchymatous organ. It is highly cellular organ, so it will be formed of two components. The first one, which will be give the outer architecture of the pancreas, which is connective tissue called stroma. The second is the functioning part, which is parenchyma. As regarding the stroma, it will be classified into three partitions. The outer one, which makes the shape, it is the capsule, which give the pancreas its shape, which give rise into septa or trabeculae to divide the pancreas into incomplete lobule. After that, there is reticular connective tissue, which is supportive in function to our parenchyma. What about parenchyma? Parenchyma, as regarding the gastrointestinal nodule, we are talking only about the exocrine portion. We say that pancreas is mixed gland, both exocrine and endocrine gland. As regarding the exocrine pancreas part, we are going to talk about two parts. The part which makes secretion and the part which deliver the secretion. The part which makes secretion will be the secretory unit, and part which delivers this secretion will be the duct system. Pancreatic secretory unit will be in a form of acini called pancreatic acini, which this pancreatic acini will secrete the digestive enzyme of the pancreas. The other parenchyma of the pancreas is the endocrine pancreas, and it is responsible for production of hormone which will be released into circulation and no need for duct system. Endocrine pancreas will be in the form of a structure called islet of Langerhans, which will release hormone. The most famous of them is insulin and glucagon hormone. So we mentioned that pancreas is formed of stroma and parenchyma. Our parenchyma will be of two parts, exocrine part and endocrine part. Endocrine part will be in the form of islets of Langerhans. Again, we say that pancreas histologically is formed of stroma and parenchyma. Our stroma is formed of a capsule. Our capsule is thin and delicate, which gives rise to a septa which divides the pancreas into incomplete lobule. This septa also will be thin and delicate. We are going to have a reticular connective tissue in order to support the parenchyma or to support both exocrine and endocrine part of the pancreas. As regarding the parenchyma, we was having exocrine part and endocrine part. Exocrine, we mentioned before, is forming of two parts, which is secretory unit and part which discharge its secretion, which is the duct system. Secretory unit was called, yes, it was called pancreatic acini, which is a serous acini and secreting digestive enzyme. Let's talk about what about the endocrine part. The endocrine part was called islet of Langerhans and producing hormone. Now we are going to talk about the secretory unit. Secretory unit is called what? pancreatic acidosis. Pancreatic acidosis is called serous acidosis according to the type of secretion. Do you remember serous acidosis? Now we are going to memorize the exocrine pancreas or the serous. We are going to start talking about exocrine pancreas, which secrete pancreatic digestive enzyme, which will be secreted from pancreatic acidosis. This pancreatic acidosis will be serous or mucus. It will be serous. So the pancreatic secretion is purely serous because we are going to have pancreatic acidosis, which are serous acidosis, which produce digestive enzyme. As regarding serous acidosis, we are going to have cell resting on basement membrane. This is basement membrane. On this basement membrane, we are going to have cell surrounding very narrow lumen. This cell is going to be tall pyramidal cells. This pyramidal cell is going to have a central rounded vesicular nucleus. Okay. This nucleus is spherical basal and active nucleus. What about the cytoplasm? What about the color of the cytoplasm? It will secrete digestive enzyme. These digestive enzyme are protein in nature. So we are going to have here a protein secreting cells, which means that we are going to have a basophilic cytoplasm with basal basophilic striation. Why it is basal basophilic striation? Due to presence of RER at the base of the cell, due to the presence of rough endoplasmic reticulum at the base of serous acidosis, which will also we are going to have Golgi apparatus and mitochondria. Our digestive enzyme will be stored in the form of acidophilic apical acidophilic zymogen granules near the apex of the cell. Do we need here myoepithelial cell in order to release secretion into the duct? No, so no myoepithelial cell here in our serous acidosis. There is another difference here in the serous acidosis. We are going to have another cell which start to appear inside. Sorry, here we are going to have our serous acidosis. Inside the lumen of the serous acidosis, there is a small cell start to appear. This cell is called in the center of the acinus, so it is centroacinar. So pancreatic acidosis is characterized by having no myoepithelial cell. What else? By the presence of cell inside the lumen of the acinus, this cell in the center of the acinus, so it is called centroacinar cells. Okay, let's see. Here we are going to have the exocrine pancreatic acidosis. Can you see here? This is a small rounded structure in this photo. Or in this photo, we are going to have a small rounded structure, dark in color. So this is serous acidosis. This serous acidosis is having pyramidal cell arranged around a central narrow lumen. This lumen contains a cell. This cell is called centroacinar cells. What about the cell? The cell is having a spherical basal vesicular nucleus, having basophilic cytoplasm, showing basal basophilic striation with apical acidophilic zymogen granules. Do we have myoepithelial cell? No myoepithelial cell. Yeah. So we missed one cell from outside and gain one inside. Lost myoepithelial from outside and gaining one cell inside the acinus, which is centroacinar cell. So can you guess the electron microscopic photo of the cells of the serous acidosis? Yes, a protein secreting cells. So we are going to have here basal rough endoplasmic reticulum, and don't miss that it is basal. We are going in between the basal rough endoplasmic reticulum, we are going to have basal mitochondria, elongated basal mitochondria. Both of them, both RER with basal elongated mitochondria, will give us basophilic striation. As this is a protein secreting cell, so with rough endoplasmic reticulum, we will also have well-developed Golgi apparatus. The secreted protein will be stored in the form of apical acidophilic zymogen granules. What is this protein? It is a digestive enzyme which will be secreted in the inactive form in order to be activated in the lumen of the small intestine. This cell will be attached to each other by junction complex. Do you know why it is connected to each other by junction complex? In order to prevent the passage of the enzyme in between the cell. What are junction complexes? There are three types of junction, which is zonula occludence, zonula adherens, and desmosomes. Let's see. This is a pyramidal cell, top pyramidal cell containing basal achromatic nucleus, basal containing basal RAR with elongated mitochondria here. Can you see this part? And this is part we are going to have basal RAR with elongated mitochondria in between. We are going to have supranuclear Golgi apparatus here, and then we are going to have the stored protein or the stored digestive enzyme, which is the apical electron dense granules, which is the stored digestive enzyme. Let's see another picture for another electron microscopic photo for this acinoid. Can you see here? Here we are going to have cells which containing apical electron dense granule, which is apical acidophilic zymogen granule. Here we are going to have a nucleus, and basal there is many mitochondria with rough endoplasmic reticulum. So this is a protein secreting cell, very characteristic of pancreatic acinar cell. By this we have finished talking about the secretory unit of pancreatic acinar cell. We talk about the pancreas. We say that pancreas is formed of stroma and parenchyma, and we divide the parenchyma into exocrine and endocrine part. As regarding the exocrine part, it was divided into part which makes secretion, which is the secretory unit, and the part which starts with the duct system, which conducts secretion, and it is the duct system. We say that pancreatic acini or serous acini of the pancreas is having a characteristic feature. It lost one cell from outside, which is no myoepithelial cell, and gain one cell inside, which is centroacinar cells. And centroacinar cell is considered the first part of the duct system. So the first part of the duct system in order to conduct the secretion is the centroacinar cell. After that, there is this small duct. This duct is called intercalated duct. So this we are coming out from the serous acini. We are inside the lobule. So we are intralobular duct. Intralobular duct join each other to form a duct in between lobule. So it will be interlobular duct. And finally, we reach to the end to open into the duodenum by the main pancreatic duct. Let's see a photo to imagine what we say. Here, we are going to say that this part was the secretory part, and we say that it is serous acini with a small cell inside it in the center of the acini. This small cell inside the center of the acini was called centroacinar cell. This centroacinar cell is considered one of the duct cells. Why? Because it makes the lumen narrow, so it helps in suction of the secretion. It acts like chalumeau. Okay, it acts like a small tube. So we are going to have a centroacinar cell which acts as the first part of the duct system of the first part of the duct system, because it narrowing the lumen help in suction of the secretion. It's considered as a cubical or low cubical cells present within the acini. Then after that it will going to form small duct. This small duct will be called intercalated duct. Intercalated duct is a very small duct. It is not prominent. It can't be appear easily. It is a small duct. This intercalated duct will join from neighboring acini with each other in order to form larger duct inside the lobule. The duct inside the lobule itself is called intralobular duct. Let's see here. This is a lobule, and another lobule. We are going to have here serous acini. Serous acini will be drained by intercalated duct to form a duct inside the lobule. This duct inside the lobule is intralobular duct. This intralobular duct will join other duct inside other lobule and join to form a large duct in between lobule. So it will form interlobular duct. Interlobular duct join each other in order to go to drain into main duct into the duodenum, which is the main pancreatic duct. Okay. Here, can you see here? This photo is a good photo. Can you see here? This is pancreas. Can you see here that there is a small duct joining each other to form a larger duct, to form a main duct which drain all the lobules. This is interlobular duct which join the gallbladder to form the main pancreatic duct which open into the lumen. Okay, so what are the components of the duct system of the pancreas? We are going to have duct system in many stages. The first of them is the cells which is present inside the serous acini. It is centroacinar cell. Then when forming a small non-prominent duct, it is called intercalated duct. After that, intercalated duct will join each other to form intralobular connecting duct. After that, intralobular joining each other to form a larger one, which is called interlobular duct. Finally, interlobular duct would join main and accessory pancreatic duct and open into the duodenum. We now have to know the function of each part and the lining cells of each part. The first part is the centroacinar and duct system, and they will make secrete alkaline-rich fluid by secretin. They are going to put secretin into the secretion brought by them under the effect of a hormone called secretin. Okay, the intercalated duct is considered as duct lined by cubical epithelial cell. Let's go to other. Now let's see other changes, other epithelial lining of other duct system. We say that we are going to have the first two parts, which is the centroacinar cells. We are going to have the centroacinar cell here inside the acini and intercalated duct. Both secrete secretion-rich alkaline-rich secretion. Why it is alkaline? In order to activate the digestive enzyme. And is it under hormonal control? Yes, it is under hormonal control of secretin. This cubical epithelium will joining together of intercalated duct will join together to form a larger duct, which is intralobular duct. Can you see here what is the lining epithelium of the intralobular duct? It is low columnar epithelial cell forming larger duct. Larger duct will be communication or joining of many interlobular duct together to form a large duct, which is called intralobular duct, and interlobular duct is lined by columnar epithelial cell. This columnar epithelial cell will open into the main pancreatic duct. This main pancreatic duct open where? This main pancreatic duct open into the lumen of part of the small intestine, which is called the duodenum. Do you remember the epithelial lining of the duodenum? Was it simple or stratified? Its function is absorption. So it will be lined by simple columnar epithelium with goblet cell. So we are going to open in the duodenum. So we are going to change our type of epithelium to be similar like that of the duodenum. So the main pancreatic duct will be like that of the duodenum. So it will be simple columnar epithelium with goblet cells because we are going to open in the duodenum. So we are going to have the same lining epithelium like that of the duodenum. This main pancreatic duct will join the bile duct before opening into the duodenum to form the common bile duct. Okay. Now let's see the function of pancreas. Now let's see the function of the pancreas. What is the function of the pancreas? Pancreas, as its parenchyma, is going to have two functions. Now we are concerning with the exocrine pancreatic function. Do you know about the exocrine pancreatic function? It means that the pancreas will make secretion which will aid in the process of digestion. So it will secrete different enzymes from the serous or the pancreatic acini. This enzyme include amylase, ribase, ribonuclease, and elastase. It will also secrete a proenzyme. What is mean by proenzyme? It means inactivated enzyme, which is trypsinogen and chemotrypsinogen. After reaching the lumen of the duodenum, it will be activated. Why? It is secreted in the form of a proenzyme in order not to digest the pancreas itself. Okay. We are also having bicarbonate-rich buffer solution. Why? There is a bicarbonate-rich buffer solution because our pancreas will open into the duodenum and duodenum receive from the pylorus of the stomach, chyme rich in HCl, so we need a bicarbonate to buffer this solution. Here, it will release its secretion through the duct system of us. The duct system starts again by centroacinar cell, intercalated, intralobular, and main pancreatic duct. So, what is the function of the pancreas? Secretion of different enzymes, including amylase, lipase, ribonuclease, and elastase. There is also secretion of proenzyme. What is meant by proenzyme? Inactivated enzyme. This trypsinogen and chemotrypsinogen, which is activated after reaching the lumen, and there is bicarbonate-rich buffer saline. Which cell secrete if the duct system secrete bicarbonate-rich buffer solution? Guess. I am waiting your answer. Which part of the duct system, under the effect of secretin hormone, will produce solution alkaline-rich solution? Yes, it is the first two parts, which is the centroacinar cell and intercalated duct. Do you remember what is the shape of this cell of the centroacinar and intercalated duct? Look at the picture. They are low cubical cells. Okay. I want to memorize you by something in the structure of pancreatic acini. Pancreatic acini was serous acini. What is the difference between pancreatic acini from other serous acini? I mean that it has no myoepithelial cell, and it contains in the center of centroacinar cell. So I have to ask you, so secretion of pancreatic enzyme is under nervous and hormonal control? Yes, it is under nervous and hormonal control. Hormonal control will come from intestinal endocrine cells, and nervous control will be by parasympathetic stimulation, which parasympathetic will stimulate secretion. What are the hormonal control from the intestinal secretion? It will be from enteroendocrine cell. Do you know the cell which controls this secretion and the quality of the secretion? The first one is cholecystokinin. What is the function of cholecystokinin? It will act on the secretory unit to make secretion. What else? We mentioned a hormone which stimulates centroacinar and intercalated cells to make alkaline-rich solution. It is secretin. Secretin will act on intercalated and centroacinar cell in order to release secretion rich in alkaline secretion. So cholecystokinin will act on which part of the exocrine pancreas? Yes, it will act on the secretory portion, which is pancreatic acini or serous acini. What about secretin? Will it act on the acini? No, it will act on the duct system. Okay. So pancreas secrete different enzyme. How does the pancreas protect itself against O2 digestion of the pancreas? I will give you a second to know how the pancreas protect himself from the O2 digestion. Yes, excellent. The first point is that pancreatic enzymes are stored in the inactive form. The apical acidophilic zymogen granules present inside the pancreatic acinar cell is an inactivated pancreatic enzyme, which will be activated only in the duodenal mucosa by enzyme called enterokinase secreted by the duodenal mucosa. Do you know one of these hormone? It is very famous one. It is trypsinogen, which will be released into the duodenum. This trypsinogen will be activated to form trypsin. Also, as if this pancreatic enzyme became activated inside the cell, God gave this cell a defense against this enzyme if it become activated. It has a trypsin inhibitor factor. So cytoplasm of acinar cell contains the enzyme in the inactivated form. So what happens if this enzyme became activated for a certain reason? Also, we are going to have another defense. What is it? It is the presence of trypsin inhibitor factor, which will prevent the action of the trypsin, so prevent autodigestion of the pancreas. By this, we are coming to the end of our lecture. Today, we discuss the changes which occur in the rectoanal canal. The landmark of us was the anal valve, where most changes occur at it. The most evident change was changing of the epithelium to come from simple into stratified epithelium and finally becoming skin. Muscularis mucosa was ending abruptly at the anal valve. Lamina propria and some mucosa after that became one layer and was containing digested blood vessel, while one internal and the other is external. As regarding the muscularis externa, the inner circular became thickened at the anal valve to form anal sphincter, and the outer longitudinal became fading and passed between the smooth and skeletal muscle. Skeletal muscle start to appear at the anal valve, forming the external voluntary anal sphincter. And we go to discuss one of the gut-associated gland, which is the pancreas. And we mentioned that pancreas is both exocrine and endocrine gland. Here in the gastrointestinal module, we are talking about the exocrine pancreas, and that is formed of stroma and parenchyma. Our parenchyma was formed of parenchyma of the exocrine part and parenchyma of the endocrine part. Parenchyma of the exocrine part was divided into secretory unit called pancreatic acini, and its secretion is purely serous because it have only serous acini. And the other part which conduct this secretion, it is the duct system. The pancreatic acini having a characteristic feature, it has no myoepithelial cell, it has centroacinar cell inside the lumen. By light microscopic, it was showing basal striation. What is the cause of this basal striation? Due to basal rough endoplasmic reticulum with elongated mitochondria in between. And then we are going to talk about the duct system, which starts by centroacinar cell, intercalated, then intralobular, interlobular, and main duct. By this, we came to the end of our lecture today. I wish you good luck and study hard. Thank you and goodbye.